So in this slide and the next one, we are going to revise again the fact of the mycelium. So the mycelium is this mass of different hyphae. So each one of them is a hypha, and the group of these threads is the hyphae, and the hyphae can be divided with septums like that, so cross walls like that, or they can be non-divided. And if they are divided with septa like that, we see this cross cell walls separating individual cells, so we call it a septated hyphae. If they don't have this structure, we call it cenocytic hyphae, or in other words, non-septated or aseptated hyphae, where you can see continuous cytoplasm and multiple nuclei in the same cell. Okay, so let's move on to the fungi reproduction. And as we said, fungi reproduce through the formation of spores. This can happen either asexually or sexually. So on the left hand side, we can see the asexual reproduction. So what you can see here, this N refers to haploid organisms. So the spores in this case, they are haploid. What will happen? They are carried by structures called sporangia. So each of these structure is called sporangium, and the plural form of them is sporangia. And these sporangia, they carry the spores. As we'll see in the next slide, we call these spores conidia. So they carry the spores, and if the spores are dispersed, that can be happen through winds, through insects, and a variety of means. And if they landed on an environment of suitable conditions, they will germinate like that, produces the hyphae, and we will have a new fungal. Once this fungal grow up, it will produce the sporangia, and again it will reproduce by spreading these spores, and so on. The sexual reproduction is presented on the right hand side of the image. This requires the mating of two different strains, or in other words, two different types of fungi. So as you can see here, the mating of one of the strains, referred to as plus, here as a minus. So since each one is a haploid, their mating will produce something like a zygote. This is a diploid structure. Then through meiosis, you will get genetic diversity. So the zygospore coming from this meiosis is genetically different in each process. Then again, it will germinate, producing a structure carrying the spores. The spores will be dispersed. If they are dispersed to a suitable environment, they will grow up, producing the hyphae, and so on. So again, highlighting the fact of fungal growth and reproduction, and we are focusing now on the left hand side of the image, where asexual reproduction is involved. So whenever fungi reproduce asexually, these spores are called conidia, and each one of them is called conidium. So remember, so these spores are called conidia, and each one of these spores is the conidium. We are just talking about the case of asexual reproduction, and the structure producing these conidia are called sporangia. So this is the plural form of the structure producing the conidium. So the structures are called sporangium. Each one is called a sporangium, and the plural of this is the sporangia. So this sporangia, they are structure that produces the conidia, the asexual spores. Once they are dispersed to a suitable environment, they will grow up producing the hyphae. And what you can see here in this image, so you see at the center, you see this dark blue or aquamarine color, and at the periphery this yellow edge. So why is that? It's because at the center we have the spores. So at the center this area is full of spores, while the edge we don't have the spores. So spores germinate from the center, and they produce these hyphae structures. And as you can see here, each spore produces this like mat-like structure of hyphae. So at the center all the spores are there, and if you blow them, you will get a puff of these spores, while the edges, they are white because they don't have the spores. Okay, so let's continue, guys, on the topic of fungal growth and reproduction. So as you can see here, that we have multiple images showing a lemon and the growth of fungi on this lemon. So it starts with a healthy lemon, and as the fungal growth progress, you can see it starts to colonize the surface of the lemon, and we see this green coloration, and as you know, they are areas dense with spores, then the white edges which are not covered with spores, and then the fungal growth extends till it completely covered the whole lemon. So how this fungal growth is able to obtain the nutrients from the media they are growing on, in this case we have a lemon, or how they are able to obtain it from any media in general, this will be presented in the next slide. So as you remember, we said the body of fungi that we see on the media is called the mycelium, and this fungal mass is built up from threads. They are called the hyphae, and these hyphae are divided into two types: the aerial hyphae, which extends in the air for the aim of dispersal of the spores, and then we have the vegetative hyphae, which extends down in the media to absorb the nutrients in a similar way to the roots of the plant. And they also share something with the roots of the plant, which is the fact that they are thin thread-like structures, extended structures, and the reason for this architecture is to maximize the surface to area volume ratio. So both the roots of the plants and the hyphae of a fungal, by having this thin structures and extended very long structures, that will ensure that we will have a maximized surface area to volume ratio, which means that they have an enhanced ability to absorb the nutrients. So this is something that fungi shares with plants. But when it comes to another strategy, we'll find that fungi has something that is very, very characteristic for fungi, and nearly no other organism share this similarity with fungi. So most other organisms will absorb the nutrients, and then once the nutrients are inside the cell, they will digest these nutrients into absorbable molecules. However, fungi do something that is totally different than that, and something that is totally unique for fungi, which is the fact that fungi secrete enzymes outside the cell. This enzyme will degrade the nutrients into digestible molecules, and then the fungi will absorb these digested molecules. It is highlighted here in this part of the figure. So here is the hyphae, the vegetative hyphae, which is extended in the medium to get the nutrients. So this hyphae will secrete digestive enzymes, and it is shown here in this part of the figure. So at the tip of the hyphae we have vesicles. These vesicles are the method for secretion of these digestive enzymes. So these digestive enzymes will be packaged inside these vesicles, secreted outside in the media. They will digest the nutrients, and then the hyphae will absorb these digested nutrients inside the fungal cell. So this is, as I said, something very unique for fungi.